Good day, this is the Professor, CBMD PhD, and today's Wednesday, August 16th, 2023. It is 7:06 and 5 seconds, AM Eastern Standard Time, and today's research, gene editing. So let's hop to it here. New gene editing tool helps zero in on small cancer-linked mutations. Date August 10th, 2023. Source: Weill Cornell Medicine. A change in just one letter in the code that makes up a cancer-causing gene can significantly affect how aggressive a tumor is or how well a patient with cancer responds to a particular therapy. A new, very precise gene editing tool will enable scientists to study the impact of these specific genetic changes in preclinical models rather than being limited to more broadly targeted tactics, such as deleting the entire gene. The scientific investigators and microbiologists genetically engineered mice to carry an enzyme that allows the scientists to change a single base or letter in the mouse's genetic code. The enzyme can be turned on or off by feeding the mice an antibiotic called doxycycline, reducing the prospect of unintended genetic changes occurring over time. Investigators can also grow miniature versions of intestine, lung, and pancreas tissue, called organoids, from the mice, enabling even more molecular and biochemical studies of the impact of these precise genetic changes. They are excited about using this technology to try to understand the genetic changes that influence a patient's response to cancer therapies. Researchers noted that differences in a single base in a gene can have functional consequences, but most gene editing tools currently available aim at larger targets, like whole genes. The scientists can also use viruses to deliver genes with specific mutations, but this technique is limited to targeting specific tissues, like the brain and liver. They've had good tools for a long time now to knock genes out or overexpress genes, but they have not had good ways to create the single base mutations that they see in patients' tumors. The mouse model allows them to study the effects of the changes on tumors and determine which therapies work best for those with a particular mutation. Organoids delivered from the mice enable detailed experiments in tissues that scientists could not easily target with the virus-based approaches. One mouse model allows you to do two things: test the effects of a mutation in cancer initiation, progression, or treatment response in mice, and take a closer look at the associated molecular or biochemical changes using organoids. As single base mutations in lung, colon, and pancreatic cancer, the genetically engineered mice will be available to other researchers to use, which may help accelerate progress toward personalized cancer treatment. They are making the technology available to other people in the field so they can use it to study their mutations of interest. If they can learn the genetic underpinnings of what causes tumor formation and why patients have different outcomes, that may help them develop new drugs or select the best drugs for a particular patient. And this is the Professor, Professor of Microbiology, Bioinformatics, Pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.